The topic that is given to me today is to talk on what's new in tuberculosis. And I have kind of thought of dividing my talk into three parts: about diagnostic, about treatment, and some future vaccinations. Before I begin, I must express my sincere thanks to my teacher, Professor Director Dr. K.C. Mohanty, the organizers of today's program, Mr. Ayamodi himself, Team Cadila, who have been working about this program for last so many months. In fact, they've been in regular touch for last so many months. All of you for having spared your valuable time to be here. Sir. This is Dr. Ayamodi, and this is the picture that I took of him at the age of 75, I think, when I met him. This is his campus, and he had the, he used to like horse riding, and I saw him riding horse early in the morning in the campus of Cadila Pharmaceuticals. If I have to, I googled about him, and I would love to share this with you, that in the independent India, when it was not thought that the medicine could be manufactured in India, he established Cadila and initiated a new era of pharmaceutical industry in country. Under his leadership, many young technocrats joined pharma industry. He was the strong hand behind Indian pharmaceutical industry's champion for Indian Patent Act, the Patency Act, and that is what has given our pharmaceutical industry the global presence. He was the one who shaped drug policy for Government of India. There are a lot of new things which Cadila has given us. Couple of things were shared: IV rabeprazole, Polycap for prevention of cardiovascular diseases, Rizorin, the molecule which is probably close to my heart, being a chest physician, the boosted rifampicin. I'm going to spend some time on it in my presentation. And also he has a credit of being cutting edge technology like VPL virus like particles for vaccine manufacturing in India. Corporate social responsibility, way back in 1985 he pioneered, and in fact he has one hospital called Kakaba Hospital somewhere in remote places in Gujarat. So many awards, and this particular stage that we have, IAMICON. In fact, whenever I read this word, IAMICON, it's like Cadila is trying to tell you, I am icon. Everybody sitting here is icon in their individual capacity. The agenda that I have in mind for next 30-35 minutes or so is to give you some overview of tuberculosis. To revise terminology, a lot of confusion when we use these terms. MDR tuberculosis, XDR tuberculosis, so-called XXDR tuberculosis, newer diagnostic, newer drugs that are available, something on vaccination, something on Rizorin that you heard about, and some take-home messages. While preparing for this talk, I looked at my last 100 patients, of tuberculosis. I have a habit of doing so. I like to look at what kind of referrals I am getting. I'm sure that would reflect the kind of practice that you have. Among the last 100 patients of so-called tuberculosis that were referred to me, when I did the workup, I realized active TB was present in 65. Only 65% of those patients were active tuberculosis. Old disease was present in 13 of them, and rest 22 were wrongly diagnosed as active tuberculosis. When I looked at what were the conditions that was thought to be tuberculosis when they were referred to me, I found out 4 out of them were interstitial lung disease, 2 were actually ABPA, 2 bronchiectasis, lung cancer, primary and secondary put together 7, community acquired pneumonia 3, upper respiratory tract infection 3, and heart failure with effusion 3. You could see that any disease could mimic tuberculosis. And amongst those active 65 tuberculosis, when I saw the kind of cases I had, the drug sensitive tuberculosis were 40, drug resistant 9, I had 28 MDR tuberculosis, I had MDR plus 12, 8 XDR, and so-called TDR or totally drug resistant were 3. In his paper, Robert Cox in 1882 said If the number of victims that the disease claims is the measure of its significance, then all disease, particularly the most dreaded infectious disease of that time, that is bubonic plague and cholera, must rank far behind tuberculosis. This is what he said 125 years ago. We all know now tuberculosis is an infectious disease caused by Mycobacterium tuberculosis, but there are so many other mycobacteria which could infect human and other living organisms. Primary tuberculosis, if I have to redefine it for you, it is the first infection of Mycobacterium tuberculosis, typically seen in children, but can also occur in adults, which is characterized in lungs by formation of a primary complex. That is, you have a small parenchymal lesion and the hilar or the paratracheal lymph node getting involved in that, which may proceed to cavitate or heal without or with maybe scarring. When we talk about relapse, we actually mean that a previously treated tuberculosis that was declared cured or treatment completed at the end of the most recent course of treatment and now again gets a recurrent episode of tuberculosis is defined as relapse. Reactivation is secondary tuberculosis that occurs as a result of activation of dormant endogenous infection, and the causes of this reactivation would include loss of immunity, hormonal changes and poor nutrition. Drug resistance could be monoresistance, that is resistance to one first-line anti-TB drug. Polyresistance, when it is more than one first line, but not INH and rifampicin together. Because the minute you have INH and rifampicin resistance together, it is called as MDR-TB. So MDR-TB is resistance to at least INH and rifampicin, the two most effective first-line anti-TB drugs. Then what is extensive? Whenever you have MDR tuberculosis plus resistance to any fluoroquinolone or any injectable, that means once you have INH, rifampicin, one of the quinolone and one of the aminoglycoside, it makes XDR tuberculosis. And XXDR refers to strain which are resistant to all first line drugs to which you can test them for. We don't have sensitivity testing for all drugs, and hence to label it as TDR is not right. This terminology is not really endorsed by WHO, but this is what we practice clinically. Look at the global burden. If this is the global burden, this is where we stand. 2.8 million of TB patients, 31% of world total tuberculosis burden is with us. Killing 1% every 2 minutes, probably by the time we end our discussion, 30-40 patients of tuberculosis would have died. Global annual incidence of 9.6 million, and we have annual incidence of 2.2 million. India, Indonesia, China, the largest number of cases, we have almost 23, that is 1/5 of the entire world burden is with us. Am I making too many movements? You could take that. I'll use this. Number of MDR cases estimated. This is old data, 2011, and this is where we stand. Same, if you go by at least one case of XDR, 2011, and I'm sure everybody here would agree that we have far more number of cases, as we don't report these cases very often. The current theory says, what happens to that Mycobacterium tuberculosis that you inhale? You inhale, it reaches the alveoli, it is taken over, kills, and the TB does not progress to disease. But when it forms a disease, it goes into the macrophage, gradually initiates a delayed type of hypersensitivity reaction. There is a caseation necrosis going on, and this would keep on increasing, extend, or it would burst that cell, would enter into the stream, that is bloodstream or lymphatics, and would have a distant spread. So that is how tuberculosis spreads. But there is a new concept which says, where we feel that when Mycobacterium tuberculosis are inhaled, it enters body directly through this M cells, which is microfold cells, which line the airway epithelium. We are not aware of this. And if we can develop some mechanism by which this mycobacteria does not come in contact with this M cells, probably we could prevent the development of tuberculosis, and that is how we are developing vaccines. So the basis of vaccines would be somewhere at this stage. I'm not going to talk about clinical presentation. So when you have to diagnose tuberculosis, you come across two clinical situations. One is latent tuberculosis and one is active tuberculosis. Latent tuberculosis means somebody who's infected with Mycobacterium tuberculosis but does not have active disease. It's like somebody having HIV has not really developed AIDS. And active tuberculosis investigations could be different. For latent tuberculosis, we always had tuberculin sensitivity testing, which is known as MANTU testing, or there are new tests available called interferon gamma. For active tuberculosis, whenever there is a clinical suspicion, whenever there is a particular history, you need to clinically examine this patient. After examination, you try to localize disease. The disease localization is done by radiology. You will get X-ray done, maybe CT done, maybe sonography done, maybe MRI done to locate disease. Once you have located disease, then you would want to confirm for tuberculosis, and there you would need bacteriological evaluation. The evaluation could be with conventional standard diagnostic testing, that is smear and cultures, or there are immunological testing, or there are new diagnostic methods like TB-NAT or BACTET, that is rapid cultures, and the newer molecular methods which could not only help in detection of tuberculosis but identification of species, but also detection of resistance to drugs, and finally give you the answer. Interferon gamma release test is mainly for diagnosing, or the quantiferon gold test is mainly for picking up the infection, that is maybe latent tuberculosis. It has got not much of clinical relevance. In fact, it creates more microscopy, gold standard. One of the most simple test, cost-effective tool, and probably 100% specific. Conventionally, we had light microscopy, then came fluorescent microscopy, and now we have LED-based fluorescent microscopy. The LED-based fluorescent microscopy requires less power than the fluorescent microscopy. It can run on batteries. Bulbs have longer half-life. They do not release any potentially toxic products if broken. No AC required, no darkroom required, and reduces lab technicians' reading time. And hence, the WHO policy recommendation says that the conventional fluorescence microscopy should be replaced with LED microscopy. So whenever there is, fluorescent microscopy facilities available, they should slowly be replaced with LED. And whenever there are conventional, they should directly opt for LED-based microscopy. After smear comes the cultures. We always had solid cultures, and solid cultures still are the gold standard. And now we have solid culture disadvantages, though they are 100% specific. The disadvantages, it takes longer time. It takes about 6 to 8 weeks for it to be positive, and another 4 to 6 weeks for sensitivity testing to be done. We have now liquid cultures, which has reduced time significantly, and you have about results coming in about 2 weeks time, and you could get sensitivity another probably in 7 days to 2 weeks time. Though by the end of 3rd or 4th week, you have sensitivity report also available. And on these machines, there are mycobacteria growth indicators available, which probably tells you about the growth, and then you could do speciation, and then you could do sensitivity directly on it. And WHO policy statement says that even the low and medium income settings should have the liquid cultures and susceptibility testing. But it says that it requires certain accredited laboratories, but we cannot be done in any laboratory. So you need BSL-2 or BSL-3 level laboratories for them to handle this kind of manipulations. And it should also have the solid cultures, because in liquid cultures there are chances of lot of contamination. Many a times we grow non-mycobacterium tuberculosis also along with it, and in times of confusion you would need solid cultures to back up your findings. After you do culture positivity, then the next thing comes is to identify whether it is mycobacteria. That means whether it is Mycobacterium tuberculosis or other mycobacteria, or NTM is the second step. Why? Because non-tuberculous mycobacteria can be resistant to first-line therapy, and there is no authentic sensitivity available for them. And hence, as you diagnose tuberculosis, your next step would be to confirm whether it is Mycobacterium tuberculosis or any other tuberculosis, and for that, you need 2 kinds of tests, or there are 2 kinds of tests available. One is conventional testing, and second is molecular testing. So what are conventional testing? We have biochemical test and antigen detection test available there. And on molecular testing, we all are aware, there are line probe, gene expert, and DNA probes available, which could identify the mycobacterium. Not only identify, but also can give you resistance pattern at the same time by looking at the mutation sequence. Capilla TB, it is one test for speciation of mycobacterium. That is, once you confirm there is a mycobacterial growth, you could do this test. It detects MPB64 antigen, and we know that this particular antigen is specific for Mycobacterium tuberculosis. Simple lateral flow speciation test. No sample processing or instrumentation is required, and sensitivity is almost 100%. So you have this kind of test available for Mycobacterium tuberculosis and Mycobacterium africanum, and your sensitivity is almost 100%. Used indirectly on the culture isolates. So either you can use it on sputum directly or on the culture isolates, and this is how microscopically observed drug susceptibility testing is done. On liquid media, microcolony detection occurs and suitable for use at reference lab by standardizing assay and testing protocol, which is available, very easily available. This is nitrate reductase assay. There is ability of Mycobacterium tuberculosis to reduce nitrate to nitrite, which is seen as color change. I don't know whether it is very clearly seen. You could see that color change here. So whenever there is color change like this, it would help you identify is as Mycobacterium tuberculosis. CRI also is a similar test where there are, whenever there is growth, there is color change, and because of this color change, you could identify them as Mycobacterium tuberculosis. And the policy statement of WHO on this is, there is sufficient evidence to use all these tests for confirmation of Mycobacterium tuberculosis and also for looking at the speciation of Mycobacterium tuberculosis. Then coming to molecular test. There is a lot of interest of late in this molecular test because it is simple, easily available, relatively cheaper, and can give you information at the fraction of time. We have PCR and we have nucleic acid amplification test called as NAT, and now we have CBNAT. CBNAT is cartridge based NAT, that is what we call it as Gene Expert. So what are these molecular tests? In microbiology, the term molecular test is conventionally used to describe test which detects the nucleic acid of the mycobacterium, DNA or RNA. All molecular diagnostic tests have three stages. There is extraction of the DNA, there is amplification of the DNA, and finally the detection of that DNA or the RNA. And that is how this is the basis of any molecular test. The near positive is of paramount importance. We do not have authentic data on smear negative samples. That means if patient's sputum is positive, probably you can subject them to this test. These tests are available the results are available in 2 to 3 days time. So in 2 to 3 days time, you can actually judge whether a patient has got Mycobacterium tuberculosis infection and whether there is sensitivity to INH, rifampicin, quinolones and aminoglycosides. You get 4 major category drugs sensitivity pattern is available. They do not replace culture positive DST. That means they are still new tests. So we should always ask for conventional culture sensitivity testing, and this should be over time probably will be more confident in interpreting these results otherwise. Commercial assays, there are various commercial assays available. That means there are simple cartridge based or simple those strip based tests available which are not really standardized yet. The advantages and disadvantages, the pros and cons. The advantage is fast tests. You get more patients on CAT for faster. If you know the drug sensitivity pattern, you know that MDR status, you know that XDR status, you could put them on appropriate treatment much faster. Otherwise, it would take 2 to 3 months before you find out about culture sensitivity pattern on liquid cultures. It may allow high throughput testing, potential for improving yield of results relative to culture. It has got very few biohazard risk and no culture manipulation. It is very standard, very automated test. The disadvantage, it may have cross-contamination risk and hence careful procedure is needed. It may fail if PCR inhibitors are available. Suppose if you send sputum and if it has got blood in it, probably it may give you false results. It is not evaluated, as I told you, on smear negative. That means if your patient is sputum negative, probably you may not get enough information on this. So you have various probes available which can help you give sensitivity towards rifampicin, INH, quinolones and aminoglycoside. 4 major group of drugs you could check the sensitivity for. The most popular test nowadays, which is Gene Expert MTB RIF. This is how the test is done. The little technical details, you have a container, you could put container on the cartridge, and that is the end of handwork. Then everything is automated, and probably in less than 1.5 hours, less than 90 minutes, probably you get a printed result which talks about confirmation of Mycobacterium tuberculosis and the sensitivity to rifampicin. And depending on the load that a particular lab has, you could put this many samples at a time. You have a simple cartridge which can take 5 samples at a time, to a bigger machine like this which can handle 1000 samples at a time and can give you result in 1.5 hours. You can imagine if you can diagnose 1000 patients tuberculosis status in less than 1.5 hours. This is cartridge based NAT, that is a CB NAT test or the gene expert test. In sputum positive, it has got fantastic sensitivity, but it has got fairly decent sensitivity for AFB negative samples also. Many times you will see your sputum is negative, but when you ask for gene expert, you get gene expert report positive. And it has got very good sensitivity for extrapulmonary sites also. In fact, of late we have started seeing its positivity in even synovial fluid. CSF and ascitic fluid we always were doing, but this even pericardial fluid we have started getting results. The experience is mounting. This is the manual NAT. I think it is not relevant now because you have automated NAT or the cartridge based NAT available, so this test probably would not be of interest. There are other loop mediated isothermal amplification. This is also one more amplification test called TB LAMP test. The indirect test for tuberculosis, antibody based test. Please refrain from using antibody based test. It has got no value, and we've got enough documentation says that this kind of tests are useless in diagnosing tuberculosis. We may have certain antigen detection tests. There are certain biochemical tests like ADA. Of late we put a lot of reliance on ADA estimation, and we feel that in exudative pleural or exudative fluid, if ADA levels are high, above 33 for pleural or maybe 9 for CSF or maybe 27 odd for ascitic fluid, it could be highly suggestive for tuberculosis. This is depending on the log of tuberculosis bacillary load per ml of sputum. Okay, so if you have the least load, the best test to ask for is liquid cultures. As probably if it is slightly more, you could pick it up on gene expert, little more LAMP test, little more probably line pro, the LPA test, because then by then the sputum is positive, and then you have a fluorescent microscopy test and the capilla, that is Mycobacterium speciation test. So you need for liquid cultures probably the least number of, even if there is minimal mycobacterial load, it can be picked up on cultures. If I have to conclude on investigations, I would say that no lab test is 100% foolproof. Phenotypic, molecular and immunological tests are no exception to that. Molecular detection of resistance depends ultimately on the presence of resistance conferring mutation. Unless there is mutation of this resistance, probably it will not pick up. It is just a mutation picking up test, and there could be alternative mechanisms. There could be alternative mechanisms of resistance. It may not be only mutation occurring in resistance development. The resistance could be because of other mechanisms as well. Mutation may appear which the test was not designed to detect. There may be other mutations which may occur, lead to resistance, and which this particular test may not detect. Hence, phenotypic tests required to identify resistance. These methods may be slow and subject to contamination, but failure in both are possible. Combination of multiple tests that include smear using fluorescent microscopy, liquid culture methods, and molecular detection to rapidly distinguish mycobacterium from non-tuberculous mycobacteria is necessary to provide reliable and accurate results for patient care. We have come a long way. For diagnosing latent tuberculosis, we were relying only on Mantoux testing, and now we have interferon gamma, we have modified Khatri test, etc. We were relying on simple microscopy, and now we have fluorescent microscopy, we have LED microscopy. We were relying only on solid cultures. Now we have liquid cultures, now we have bacterial cultures, and now we have a lot of molecular diagnostics available for diagnosis of tuberculosis. But still, no testing method can replace clinical assessment. Many a times all your tests would fail, but your clinical suspicion would make you suspect tuberculosis, and microscopy and culturing still would remain on top priority. Coming down to treatment for tuberculosis, we all know the category 1, category 2 treatment for tuberculosis. But when it comes to MDR tuberculosis treatment, there is a grouping given for drugs. Group 1 drugs is the first line drug, that is INH, rifampicin, ethambutol and pyrazinamide. Group 2 drugs are the injectable anti-TB drugs, that is streptomycin, kanamycin, amikacin and capreomycin. Group 3 drugs are fluoroquinolones, that is moxiflox, getiflox, levoflox, ciproflox. Group 4 drugs are oral second line anti-TB drugs, that is ethionamide, prothionamide, that is cycloserine, terazidone and that is PAS. And group 5, that is drugs with unclear tuberculous activity, but we still use it when we have no other drugs available, that is clofazimine, linezolid, amoxicillin clavulanic acid, thiacetazone. We know that imipenem also has anti-tuberculous activity, high dose INH and clarithromycin. In fact, the recent papers, the recent studies say that probably clarithromycin has got no effect and that is not considered as the group 5 drug anymore. If you look at the historical timeline and discovery of TB drugs, we started and this is what we are. And the way we are treating our tuberculosis probably and the way the newer diagnostics are going to help us treat tuberculosis, we can be sure that by 2015, we will be able to, if not completely eliminate TB, would be able to control TB to a major extent. Prevention is definitely better than cure. New drugs. Since the rifampicin, there is not a single new drug which was specifically designed for tuberculosis hit the market. There are 2 new drugs which recently came, and that is bedaquiline and delaminid. These are the 2 drugs which are approved by appropriate authority in US and Europe for conditional use. It is conditionally approved. That means it is not for anybody's use. In a specific condition under supervision they can be used. And bedaquiline for MDR, it can be used. Bedaquiline for MDR and delaminid is only for compassionate care for XDR and TDR tuberculosis. So it is not to be used for MDR tuberculosis. As of now, we can use it only for XDR tuberculosis. This is the drug profile of that. It is diarylquinolone class of antibiotics, selectively targets proton pump of ATP synthesis, approved only for MDR tuberculosis, 400 milligrams daily for 2 weeks, and then the dose needs to be reduced to 200 milligrams 3 times a week for next 22 weeks. That is total 24 weeks treatment only. And delaminid is nitramidazole class of antibiotic, inhibits the synthesis of mycolic acid, approved for MDR plus actually tuberculosis. There are so many newer drugs in pipeline in the diarylquinolone group, DRP-E1 inhibitors, MMP-L3 inhibitors and oxazolidones. In fact, we have used oxazolidones. We have been using linezolid in tuberculosis, and then sultazolid is already available. I think it's just the time when it is available for use, but these are the drugs which we will be soon seeing. These are the promising molecules which are in pipeline, which are in various phases of clinical trial. These drugs probably has entered phase 1, this is in phase 3, and there are 2 more molecules which probably we would see in time to come. WHO recently updated their guidelines and their class-wise distribution of drugs and have put add-on drugs into 3 categories, D1, D2, D3, but I'm sure this is only for programmatic use, and in time to come there will be a lot of restrictions on treatment of MDR, XDR tuberculosis in private practice, and probably we may not be at the liberty to treat tuberculosis like this. Updated new guidelines say that probably the duration of treatment will come down from 18 to 24 months to about 9 to 12 months, and shorter MDR-TB regimens would be recommended under specific conditions only. So if you look at composition of revised MDR treatment, which you're going to see in time to come, rifampicin resistance has to be there for it to be labeled as MDR tuberculosis. In the last part of the presentation, when we realized that there are no more medicines available, rifampicin was only the last anti-tuberculous drug which was specifically developed for tuberculosis. Cadila Pharmaceutical at Ahmedabad developed a newer formulation of rifampicin to overcome the limitation, and it is boosted rifampicin, what they label it as Rizorin. Rizorin is actually the combination of, combination of rifampicin, INH and piperine. Rifampicin against the conventional dose of 450 is given as 200 milligrams. INH remains the same, that is 300 milligram, and piperine 10 milligram is put into it. Why this combination? What is the rationale behind this combination? There are certain studies or certain understanding of rifampicin metabolism. There are thoughts that rifampicin probably leads to auto-induction of cytochrome 450 enzyme. So what does it do? It leads to probably, if I have to put it in the word, apne pair pe kulhadi marna kind. When rifampicin is given over regularly over time, it lowers its own bioavailability. In the sense, it leads to overexpression of something called as P-glycoprotein, which is actually an efflux pump. That means it effluxes those medicines from the intracytoplasmic, from within the cell, and it leads to cytochrome 450 expression, which leads to reduced bioavailability of rifampicin. There are studies which say that if rifampicin and INH are given together, INH reduces the bioavailability of rifampicin. Stability of rifampicin can be enhanced only if the INH is coated. That means if a coating or to INH is given and then it is prepared, then only probably the rifampicin bioavailability is not altered. There was an observation made in the areas around Jammu that whenever patient received rifampicin of 450 milligram, they had lot of GI intolerance. And when they were studied further, it was realized that the piperine content of the diet is little high, and that makes rifampicin more bioavailable, and they tried giving less of rifampicin and saw that it gives comparable results, and that was the rationale behind combining this as a bioenhancer. So what does this bioenhancer do? Piperine increases the bioavailability of this drug through inhibiting cytochrome 450. So what does rifampicin do? It overstimulates it, it inhibits it, so it's kind of balanced to this cytochrome 450, and it also inhibits P-glycoprotein, which is also responsible for auto-regulation of rifampicin. And hence, rifampicin being potent inducer of cytochrome 450 and P-glycoprotein, piperine is a potent inhibitor of that. So there is one stimulator and one inhibitor. It kind of bring about the balance, and hence the inhibitory effect of piperine compensates the inducing effect with rifampicin, making rifampicin more bioavailable. With that, and in that, the INH is given into coated form, so it does not really hamper the bioavailability of rifampicin. They have done a lot of bioequivalent studies in-house, and it compares that rifampicin is the rizorin, which is having 200 milligrams of rifampicin with piperine, is bioequivalent to 450 milligrams of rifampicin otherwise, and it is definitely better than 200 milligrams of rifampicin standalone. That means if you give rifampicin without piperine, it would not serve the purpose. Unlike with conventional rifampicin, the blood levels of rifampicin do not fall with continued use. That means there is no auto-regulation of rifampicin happening over continuous use of rizorin. There are various clinical studies which are done and which show, in fact, I have a little experience of using this kind of combination. I have used it in patients who could not tolerate rifampicin. That means they had GI intolerance, or they had mild fluctuations in their SGOT, SGPT, and they were symptomatic for that. In those patients, we have used little lower dose with piperine, and we found comparable results. Bcg vaccine, we all know that the pros and cons and the advantages and disadvantages of Bcg vaccine. There is a school of thought which says that Bcg vaccine is completely useless, and there are so many pediatricians which refrain from recommending Bcg vaccination, versus we feel that Bcg vaccination should be given. And there are various subunit vaccination which are the potential candidates, though none is really hit the market. But then this is what is the pipeline for newer vaccines, and I'm sure in time to come we would see these vaccines as well. To conclude, I would say no tests are 100% reliable to detect tuberculosis or to test drug sensitivity. We may have to combine a few tests with our clinical impression to arrive at a conclusion. Newer drugs are few, and we may have to learn from a past experience, and we have to restrict its use. Vaccination, yes, it will be available, but has a long way to go. Thank you. Thank you very much for your patient hearing.
was really amazing and really insightful. So if you can so do we, for the audience, do we have any questions?
You want to take a question now? If there are any questions, any suggestions, any difference of opinion? Oh, for tuberculosis, nobody has any queries? Such a common disease, across specialty we see that.
In the protocol, for a surgical specimen, how many days we should perform? Because during time, you cannot get the surgical specimen repeated to repeat the test.
True.
What is the protocol one should follow? What are the tests you should follow?
That's an excellent question. And hence probably no other tests got that kind of light. So I think, so far as current scenario is concerned, gene expert probably would be the test to ask for. So you got a question?
I wanted to emphasize that you should, as told about TB being a notifiable disease, and it should, everybody treating TB should compulsorily notify.
That's an excellent point. Everybody must notify tuberculosis. And in fact, good you got that point out. It is available as a, you have application on mobile. You could download that application and you could just, put patients, there are very few details that one needs to fill in and it gets notified. I think that's one step we could do to help get better idea of disease in our community.
Gold test is discarded.
Sorry, sir?
TB gold test.
TB gold test is, no, it is not discarded, but it creates more confusion than, benefit. And I don't think that we are interested in knowing about latent tuberculosis anymore. We are yet to look after our active cases. The finding out of latent tuberculosis doesn't really solve the issue.
Incidence of blood resistance is very high. Are these figures of KG Somaya or your hospital? Exactly from where?
This is just my figures. This is my last 100 patients. I have a habit of looking at my last patients, And being in Bombay, in a tuberculosis setup, probably my figures are definitely going to be higher. It's a specialty clinic, so probably it can be. Obviously, sir.
Yeah.
Obviously.
Highest incidence is you are working in a place where the incidence of TB is very high.
No, sir. What has happened is the first line tuberculosis doesn't come to you anymore. It is treated at RNDCP centers. If you really ask me how many community-acquired pneumonias I saw in last 6 months, I would say probably 5. And at a general practitioner level or at a physician's level, probably they would be seeing 10 times than what I see. Because community-acquired pneumonia is always gram-positive, and whichever antibiotic you use, it gets cured. So as probably, those TB does not come to us. So when there is complication, when there is resistance, only that probably tuberculosis gets referred to chest physician. So those figures probably would be entirely fine.
What is the relevance of dots now?
Very good. Sir, imagine, we are talking about Pune and Mumbai. Imagine we have to design a program which caters to entire country. I was the last, city to get RNDCP was Northeast. I think from Itanagar I went somewhere. Mr. Dorji Khandu was the Chief Minister. I forgot the place where I went. That was the last city to be enrolled into this RNDCP program. Imagine, sir, anywhere in the country you stand and in 1 kilometer you have access to anti-TB drugs. Fantastic program, isn't it? We are not talking about individual patient management. We are talking about community management. We are talking about mass management of tuberculosis. You imagine if somebody gets completely free anti-tuberculosis treatment for MDR and XDR tuberculosis, that's a big boon. We understand there are some issues. We understand we need to learn a lot. But imagine CB-NAT, gene expert done completely free of cost. Sir, a test would cost you easily 2,500 to 3,000 rupees, right? If it can be done. There are pros and cons, but overall, if you really ask me, it has done wonders.
Dot has been discarded by WHO.
Sir, it is dot nahi. It is the intermittent chemotherapy. Yes, intermittent. We realize that probably intermittent chemotherapy doesn't work as well, and we need to change to daily regimen. So dot is not scrapped. The intermittent regimen probably is scrapped. So we would be soon putting patients on daily regimens.
How do you treat MDR and XDR?
Sir, again, it depends on what kind of disease it is, but average feeling is about 18 to 24 months, and you need to monitor them on cultures. So once your cultures are persistently negative, disease is absolutely quiet, and probably, for 2 years is that. We were treating for 1.5 years, now we realize that 2 years is what we need. But the newer treatment which we are going to get would bring it down to again 9 months to 12 months.